It's an honor to be here. I can remember very distinctly when I heard for the first time the story about how you guys were curing lupus, which is a serious disease for some people, with a single shot. It blew me away. And I was like, that paper, the Nature Medicine paper, I mean, I sent it around to all my friends, colleagues, saying, you know, this is one of the most important advances in the business. Unfortunately, I think it came out during COVID, so people were preoccupied. But I still think it's amazing. And every time I hear the story and what's happening with the antibodies and the B cells, it's just fascinating. Unfortunately, I'm not here to say that we're making the same kind of progress with HIV. It's going to be challenging to use CAR T cells for the cure of HIV, but we're trying. So let me just tell you the story. First and foremost, right, we all know that HIV is a treatable disease, right? In the mid-1990s, we developed these combination regimens with multiple pills. They were quite toxic, but they sort of worked. But now we're up to a point where almost everybody who has access to therapy and wants to take the drug and can tolerate it can get by with one pill once a day, with probably a normal lifespan. And within a year or two, we're probably going to be able to do injections every 6 months. You know, and so the real question is, why are we even thinking about something as potentially expensive and challenging as CAR T cells for HIV? Well, because it's actually a lot of unmet needs with antiretroviral drugs, and it's complicated. A lot has to do with, you know, how people experience taking lifelong therapy. A lot of it has to do with public health issues. People on long-term therapy still suffer from stigma, discrimination. These medications that we're given, although generally okay, have been associated with weight gain, metabolic abnormalities. And, you know, a lot of people that I take care of have multi-drug resistance, and they're not able to respond to these therapies. That's the individual level. But the big issue is the public health stuff. You know, HIV is a global disease. Most of the burden's in Africa. We're doing pretty good now in terms of getting drugs to people, but it's always a challenge. Most of the therapy that we deliver now in many countries is funded by PEPFAR, the United States government plan, which came this close last month to being discontinued, which would have huge impacts on the ability of disseminating therapy in Africa, and so forth. So there is now kind of a global effort to come up with basically what you guys are doing with lupus and other autoimmune diseases, a one-shot cure to basically take care of this virus once and for all. And that's where our group is working. So let me just give a background, a little bit about the challenge here. HIV integrates within CD4 T cells. Infected cells are relatively rare. Only about 1% of cells, 1 in 1,000 cells actually harbor the virus during treatment. Most of the viruses that you see in infected cells are actually defective. They can't replicate. They're either mutated, have deletions, or they're stuck in parts of the chromosome where they can't get out, right? The sort of the graveyard area of DNA. Which means the virus that we care about, you know, the virus that matters, the virus that can rebound, is actually quite rare, hard to detect, hard to measure. But it's there, and it persists indefinitely. And so the question is, in terms of cure, can we target that small, very small, difficult to identify reservoir? Can we target and get rid of it, which is the cure, or can we come up with ways to control it? Okay, and so these are the two big efforts that are going now for HIV cure, either total elimination or allow people to go off therapy, have the immune system control the virus. And so the question is, what role will CAR T cells have in this process? All right, so 1 in 100 people who acquire HIV control the virus on their own for basically their entire lifetime. They're called elite controllers. And everything we know about these individuals is that 90% of what's happening in them is CD8 T cells, right? CD8 T cells that have the capacity to proliferate, target very specific areas of the virus, and persist if you have all that, you know, if you're born with that, it's probably it's mostly genetic. Then when you acquire HIV, virus will replicate for a couple of weeks, it'll come back down, and you're good for life with the immune system controlling it. So this has led to really 25 years of clinical trials using vaccines, adjuvants, cytokines, to sort of come up with a way to turn on these CD8 T cells so they can control the virus. And in my opinion, with rare exceptions now with what's happening in some of the antibody work we're doing, all these approaches have failed, right? We have not been able to achieve any real degree of CD8 T cell mediated control with vaccines and vaccine adjuvants and PD-1 and so forth. And so that gets us to CAR T cells, which is an interesting story. A lot of the original work on CAR T cells occurred for HIV back in the early 1990s at the NIH, the Intramural program. Carl June at the time was working on CAR T cells. We didn't call them CAR T cells then. He essentially put together a company called Cell Genesis with a bunch of other colleagues in San Francisco. I was a young physician at the time. I'm not a young physician anymore. This was 30 years ago. They basically said, you know, we have these magic cells that can actually go throughout the body and kill the virus. What would you do? And I said, well, let's put people on antiretroviral drugs. And at that time, we were beginning to realize that most of the virus was in the gut. Let's put people on therapy and then give them these cells that you guys are making, see if they can go into the gut and get rid of the virus. And we did this study. It was complicated. It was first-generation CAR T cells, no co-stimulatory molecules or anything like that. The CAR was CD4, which bind directly to the HIV. And it looked like it reduced the amount of virus in the gut, but the p-value was 0.07. And by that point in time, this is now late 90s, everyone realized that, that we don't need these cells for HIV because these drugs were getting better and better. One thing that we learned from this, and this is again work that we've done from that original clinical trial, is we're continuing to follow these guys. So this has now been, you know, 20, 25 years. And last time we looked, and this is work that, that was published in Science Translational Medicine a few years ago, these cells persist, pretty stable. Again, this is without all the co-stimulatory stuff. These are just first-generation CAR T cells. They persist in a stable way for years and years, presumably decades. Now, whether they're doing anything is hard to clarify. But we continue to follow this cohort. There were no safety concerns at the time. We weren't aware of cytokine release or neurotoxicity or T-cell cancers, and so forth. But what happened was we did a handful of these studies. There were 4 or so back in the 90s. Then the entire CAR T-cell world left us. They just took off. We said, okay, we're not interested in HIV anymore. We're going after cancer, which made great sense. So we said bye-bye, and the entire CAR T-cell work moves into cancer, where great things have happened. And now, 20 years later, there was a big break. That same group, many of them are now coming back to the HIV world to see if we can actually recapitulate now with the much more advanced technologies, whether or not we can actually achieve better outcomes with the modern day CAR T-cell studies. The difference between 1996 and 2024 is that the risk-benefit analysis of what we can do in people with HIV is fundamentally different, right? Back then, we thought HIV was basically a fatal illness, and so we were doing all sorts of cutting-edge, innovative, high-risk stuff. Now it's all about safety, 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 because most people, not everybody, you know, can basically access therapy, go on drugs, and do quite well. All right. So where are we now? So now we're basically getting to the point where we can now begin to think of what a cure might look like. And a lot of the interest in cure is actually being driven by these bone marrow transplant cases. The first one, which Berlin patient, was actually done here in Berlin by Gerald Hutter and others, in which we used, he used a donor who lacked CCR5 and did an allogeneic transplant to a person with HIV, Timothy Brown, the so-called Berlin patient, very famous in our world, a person who actually became my patient for about 5 years, Timothy Brown. So I followed this case very carefully. But since then, we've had at least 5 cases in using using basically cell-based therapy to cure HIV. Okay, but this is allogeneic stem cell transplant with all sorts of morbidity, not something you do in someone without cancer. But it's proof of concept that using these cell-based therapies, we can achieve a cure. There's also an interesting story emerging that there are so-called natural cures. There are at least 2 women we know of globally. One's, again, from San Francisco, a patient we follow, and another is actually from Argentina, who acquired HIV and cured themselves, right? I mean, we cannot find the virus anywhere in both of these individuals. And at least the San Francisco person has very powerful, potent CD8 T cells. So anyway, so we're making progress in terms of achieving a cure, but nothing so far has been scalable. Now, as I begin to show you what we're doing with CAR T cells, there are 2 therapeutic approaches one can take, all right? You can either, as shown here, people go on antiretroviral drugs, and they basically, whoops, what did I do? Okay. What you see in this figure is that if you go on antiretroviral drugs and you stop therapy, 7 days later, the virus begins to spread, okay? Begins to replicate in the tissues. A lot of stuff happens in that first 4 or 5 days when the virus spreads. And whether if the immune system responds effectively, then you can control forever. If it doesn't, then the virus actually wins and takes off and you can never catch up. So it's possible that if we have the right cells, let's say CAR T cells that are HIV, that target HIV, in the right place at the right time, transiently, just that key intercept, then you can totally change the future of HIV. So that's kind of what we're doing now in our clinical trial, is identifying a way to give these cells so they're there at the time the virus begins to rebound, to basically push the virus in the direction in which the immune system controls. Or you can think about the fact that you just need long-term CD8 T cells forever and ever and ever to control HIV. Obviously, very different approaches. Both are testable now with the techniques that we're using in the clinic. So what are we doing? So here we get into some of the CAR T cell biology, and I'm going to say right up front, this is not my thing. I'm a clinician, clinical investigator. I use the tools in people, but I don't develop them. But this was developed by Kim, Anthony Gonda, Boro Dropulik, and colleagues who are now at Charing Cross. And what they did, and this was published in Science Translational Medicine a few years ago, but they basically used a lentivirus to develop a dual CAR that when expressed binds the CD4 binding site and the CCR5 binding site. So importantly, it identifies 2 epitopes on the HIV, and it binds to that with a co-stimulatory molecule as outlined here. And so essentially leukapheresis is done, cells are sent to Cleveland, the T cells are modified, CD4 and CD8 T cells, so they're sent back, and then we put them into people, which is my job. In terms of some of the more clinical data, there's been lots of preclinical observations made with these cells, but Kim's work showed that in a humanized mouse, when you use 1 or 2 of these constructs, basically they both work. Either there's identify 2 binding sites on the HIV or 3, both work fine in terms of clearing HIV in this humanized mouse system. So this provided the rationale and the proof of concept to do the clinical trial that we did. All right, so we got an IND to do this study actually a few years ago now. And I'll say right up front that implementing this study in people has been extremely challenging. Again, the risk-benefit analysis of going to people with HIV is difficult. Even though globally there are this great unmet need, terms of people accessing therapy and staying on therapy, the people who go in the clinical trials are generally the people who are doing well. And so there's a big difference between the target population and the people we're treating. The people we're treating are generally on treatment, doing well, normal lifespan. So you have to be very, very careful in terms of balancing everything. And so therefore, we're starting off with low doses, not using conditioning, all sorts of monitoring. And implementing this at UC San Francisco, where I work, proved basically impossible, in part because of all the amount of cutting-edge cell-based therapies that are being done now in cancer. So essentially, there was no capacity at UCSF to do this. So we're doing this study now in collaboration with colleagues, Mehrdad Abedi and others at the University of California, Davis. It's just where, again, it took a while to get this thing up and going. So what's the study look like? It's a traditional dose-escalating clinical trial, first in human. Primary objectives were safety, 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 but we're also looking at persistence of the modified cells, as well as the capacity of these cells to control HIV during a treatment interruption. Study design, cohort 1, again, these are people on long-term therapy, high CD4 counts, no comorbidities, relatively young. Cohort 1 got a low dose of the cells without any conditioning. And then these people were enrolled sequentially. We looked at the safety. And then if there were no safety concerns, we went on to cohort 2, where we are now. And in this situation, we're using cyclophosphamide. I think it's about 1 gram, so low dose of cyclophosphamide given as conditioning on the day of the infusion of the T cells. And then, again, low dose CAR T cells. And then once we get through three of these people, we'll go basically with dose escalate the number of cells. And there's actually a 4th cohort in which we'll give the cells twice during the interruption period. So again, things have been going slowly because of all the bells and whistles in terms of doing the study, some of the capacity issues. And so right now we're basically into cohort 2. So here's what happens in people who interrupt therapy, okay? So we don't have a control arm. So it's important to understand that we really do understand pretty precisely what happens when people stop antiretroviral drugs. The virus is suppressed, it's undetectable, you stop therapy, the drugs leave the system in 5 days. Day 7, we know this because we've done really intensive work here. Day 7, the virus begins to replicate in the tissues, and around day 10 to day 14, the virus becomes detectable in the blood, and then within a week, it's up to 1 million copies, okay? There's some distribution in terms of time to rebound. Most people rebound at around day 10 to 14. Some people can wait up to 3 weeks. But for the most part, whether you start therapy early or you start therapy late, basically it's a pretty consistent pattern. So we know this from doing these studies over and over and over again. So keep that in mind as you look at the data that we're generating so far in the context of this clinical trial. So this is the first person that we've studied, let's see. So the red here is the viral load. So here's day 0, no conditioning, low dose of the CAR T cells. Virus actually was blipping at very low levels, undetectable but detected, and then rebounded around day 21 was the first time we saw the virus. Actually, the first time we saw the virus in the level that we thought was real was here. So it was over about 3 weeks, which is longer than expected, okay? But, you know, this does happen, so can't make too much of it. Again, most people rebound around day 14. So maybe there was a bit of a delay. This is the CD4 counts, didn't stay. And once the virus took off, it basically, the kinetics were as expected, and the person went back on therapy. And so in terms of the cell persistence, this is the CAR T cells. The infusion was done here. I think you get this pretty typical increase in the number of cells that were circulating. Then it actually dropped and slowly went down to levels that was no longer quantifiable. In the second person, for which I have data to share, this person also actually, so the red here is the viral load. And again, first time that the virus actually was detectable was almost 4 weeks. Again, longer than expected. So maybe there's a hint that the CAR T cells, low doses, are actually delaying rebound. But again, can't make too much about this early days. CD4 counts remain stable. Of interest in this person was the fact that, that the cells went up as expected, came back down, but looked like it blipped right around the time the virus came up, suggesting that the virus was actually able to induce expansion of the CD8 T cell, the CAR T cells in vivo, something we weren't sure was going to happen. So that's encouraging if in fact it's true. All right, so this is, these are the only 2 people for whom we have complete data on. We've actually enrolled now 4 people. The 5th and 6th are actually pending. We're into the second cohort, as I said, with the cyclophosphamide, and we'll see what happens. So just back up a little bit and talk about some of the concerns that we have in doing this, how we think things are going to go forward. This was a very nice paper that was reviewed yesterday about the potential complications. Most of these apply to people with HIV. Let me just go through them real quick. One question that's always been out there is that people with HIV, PWH, you know, their immune system, their CD4s are normal, but their immune system is persistently abnormal. There's chronic inflammation and so forth. And so the question has always been whether or not CAR T cells are going to function in that environment. All we know is that ex vivo, when people have these CAR T cell modifications, they appear to function as expected. But this remains a concern, that the persistent immune dysfunction is actually resulting in issues in terms of whether these cells will work. The big problem that we're dealing with is not enough antigen, right? This number of cells that express HIV is rare. The amount of HIV on the cell surface is hard to detect. And so it's not entirely clear that we're going to be able to target HIV-infected cells with current approaches. Now, what are we doing? There's a big, huge effort to reverse latency, basically give people drugs to turn on the virus so it comes out so the immune system can see it. Some progress is being made. So in theory, CAR T cells are going to have to be administered with things that reverse latency. We're working on that. The other thing is to optimize the number of CAR T cells, their function, their activity, so that when the virus begins to rebound and there's lots of HIV around, that's where the CAR T cells are working. That's what we're trying to do. And of course, there are new CD8 T cells, new CAR T cell technologies that are designed for these low antigen states. We'll see how they work. Escape is a big problem. HIV can mutate and evolve very quickly, and this has been an issue that we've dealt with. You know, and that's in partly why we're using these dual CAR T cells, the combination therapy approach. Toxicity is a main area of concern for us because of the risk-benefit ratio in our patient population. I think it's more significant compared to the lupus stories, the autoimmune stories, certainly cancer. The ones that we've concerned about initially, we think are not going to happen, again, because the amount of antigen around is so low. But we're monitoring very carefully. We've done a bunch of these. We haven't seen anything in the CAR T cell studies we've done. The other group that's done a lot of CAR T cells is Jim Riley, Carl June, Pablo DeBas at Philadelphia, at Penn, and they've not seen any toxicities in their cells, in their studies to date. You know, and the issue of T cell cancers is a big one for us. We're really concerned about this. Obviously, we go through this carefully in the informed consent process. I understand there's a fair amount of debate going on to what degree this is actually happening. I know that some of the other gene therapy approaches that we're working on in HIV are going to use site-specific editing to basically insert the gene in areas that we want it to go, the safe harbors, in the CCR5 gene, for example. There's discussion about using non-integration approaches. And then discussions here yesterday about using technologies in which the amount of CAR expression is actually transient, which might help us, particularly if we want to go after the rebound period, might also be a way to go to achieve short-term control until the immune system can take over. So this is sort of some of the areas of interest going forward. Let me just end with this anecdote. So this is actually from the University of Pennsylvania group, Pablo Tabas, Jim Riley. As I said, they've been doing a lot of cell-based, gene-based therapies, knocking out CCR5 with gene editing and so forth. They actually have this one person, which I find fascinating, that they've not published yet, but they've reported at a bunch of meetings. This is an individual who received 2 doses of ex vivo editing of CCR5 in a subset of cells, which by itself doesn't do much, but they knocked out CCR5 in many of the CD4 cells, not all of them, so they're immune to getting infected with HIV. And then on top of that, they got a CAR T cell approach using CD4 as the binding, as the CAR, basically the same CAR that we used back in the 90s. And basically they showed that once with the CCR5 editing alone, the person rebounded. But later on, after having gotten 2 doses of CCR5 editing and now the CAR, the person hasn't rebounded. And that actually conceptually is very cool, because 2 things are happening there. You're giving the CAR T cells that control the virus, but you're also making the virus work harder to replicate because there's less target cells. And that's kind of conceptually how we're thinking about this whole HIV cure thing. You want less virus and better host immune system. So that's just sort of an anecdote that I think is providing some inspiration for what we're up to. Just finally, just one last slide, one last story. I just want to just call out this work that Boro Dropulik and his team is doing at Caring Cross. It's very inspiring. We have a state of California CERM grant to do this, and a big part of that grant is to develop the capacity to expand access globally to people with people who are in need of CAR T cell therapies. And I'm not going to go into any details, but they have all these technologies. They have all this intellectual property that they can basically pass on. But with the grant that we have, we're trying to implement this approach, particularly in sub-Saharan Africa, where most of the burden of HIV exists, and where we think we can get the cost down to a level where it actually becomes financially feasible to even think about using these approaches in a place like sub-Saharan Africa, which obviously right now is not going to happen, considering the cost, but there is this effort to basically begin to think about how to expand these expensive approaches. Again, it's not my area of expertise, but it's part of the program that we're running. So I'll end there. Again, thank you. I really thoroughly enjoyed yesterday's discussion. I like being in meetings with a bunch of people I've never met before, doing things that are really cool, and having great discussion. So great, thank you.